Hello, AP Biology students. We're going to go over the fifth section in Unit 5. And in this section, we're going to talk about something called genetic recombination. Up until this point, we've been talking about how Mendel's data started our understanding of the world of genetics. When Mendel published his data, he wasn't given notoriety for the understanding of how traits were passed on from parent to offspring. Basically, it took years for science to catch up to how important his data was. Two scientists that really showed how far ahead Mendel was were Baveri and Sutton. They were the first to really show that chromosomes are how traits are passed from parent to offspring. And something within the chromosome's DNA is going to be specific for that transmission. Specifically, in this case, it would be the genes. Another scientist that was influential in this understanding of genetic inheritance was Thomas Hunt Morgan. He's a scientist that studied fruit flies and analyzed the frequencies of certain traits in the offspring. And when he was doing his experiments, the ratios that he was getting just didn't make sense to him. In Morgan's experiments, he made the hypothesis that he should see the same frequencies play out in his fruit flies as Mendel saw in his pea plants. Specifically, the outcomes of a cross between two parents should see an even amount of each chromosome being inherited, that being the 25%, 25%, 25%, 25% that we see here. However, what Morgan saw was a 48%, 48%, and then 2% and 2% for the outcomes. And this didn't make sense to him. Those numbers were way too far off just to be, naturally occurring in nature. So he was perplexed. He didn't know what was going on here. Why were these percentages not looking like the ones that Mendel saw? Morgan was a professor at this time and actually posed these questions to his students. And in his class, there was a student named Alfred Sturtevant. And the story is that one night he blew off his homework to try to figure out why these ratios weren't working correctly. And basically what Sturtevant did was he figured out what crossing over was. Remember that crossing over is the genetic exchange between non-sister chromatids of homologous chromosomes. And this process specifically occurs in meiosis 1. Sturtevant realized that the genes that Morgan was looking at were on the same chromosome. Remember, the term locus refers to the location of a gene on a chromosome. So what Sturtevant was looking at was, where are these genes specifically located on the chromosomes? Morgan initially thought that these two genes were on separate chromosomes, which would have led the chromosomes to be independently inherited from each other. And this is why the outcomes should be an even 25% for each of these gametes. What was really going on was these genes that were associated with the traits were on the same chromosome. Since they're on the same chromosome, this led to the inheritance of these genes to be different than that 25%, 25%, 25%, 25%. This is why they had this 48% and 2% in the outcomes. This led to the understanding of something called recombinant DNA. This is when chromosomes are genetically different because of something called that crossing over. You can see crossing over occurring between these two homologous chromosomes, and each chromatid in these pairs of homologous chromosomes is genetically different from each other. This leads us to an important aspect of looking at how genes are going to be inherited. This is linked versus unlinked genes. Linked genes are genes that are located on the same chromosome. The closer two genes are together, the more likely they are to be inherited together. So you can see this cluster of genes here are linked because they are located so close together on a single chromosome. Unlinked genes are genes that are on different chromosomes. As you can see, this gene 2 and gene 1 are unlinked genes because they're on different chromosomes. Now, this doesn't mean dihybrid crosses are useless, but we have to make sure that the genes that we're associating with the dihybrid cross are unlinked, because we want to make sure that independent assortment is going to be the sole reason why we see those ratios and not just crossing over. When it's crossing over, those ratios won't work out.